Hello viewers, welcome to my channel Biology Notes. Today I will discuss about the intramembranous mode of ossification or osteogenesis or bone formation. First of all, we must know what are bones. Bones are lining tissue which have their own blood vessels. Bones are made up of various cells, proteins, minerals, vitamins, etc. The bones provide the structure of our bodies. The adult human skeleton is made up of 206 bones. The bones may be of various shapes and sizes. So, it is very important to know how the bones are formed. There are two methods, intramembranous and endochondral. So, in this video, I will discuss intramembranous mode of ossification. In intramembranous ossification, bone formation is initiated by condensation of mesenchymal cells. These cells then differentiate into osteoblasts. The flat bones of the skull, face, mandible, clavicle develop by intramembranous ossification. The first evidence of intramembranous ossification is seen around 8th week of human gestation within embryonic connective tissue, which is known as mesenchyme. Here I have tried to discuss the process. Suppose in the mesenchymal tissue, these are mesenchymal cells and these are collagen fibers. The mesenchymal cells are spindle-shaped and pale staining. Some of the mesenchymal cells migrate and aggregate in specific areas. These areas are known as ossification centers. This condensation of cells within the mesenchymal tissue initiates the process of intramembranous ossification. Mesenchymal cells in these ossification centers elongate and differentiate into osteoprogenitor cells. Now, these osteoprogenitor cells express CBFA1 transcription factor. This transcription factor is essential for osteoblast differentiation and the expression of genes which are necessary for both intramembranous and endochondral ossification. Next, osteoprogenitor cell cytoplasm becomes basophilic and a clear Golgi area becomes evident. As a result, osteoprogenitor cells differentiate into osteoblast cells. These osteoblast cells are important for synthesizing bone matrix and coordinating the mineralization of the skeleton. So, osteoblast cells then secrete the collagens, bone sialoproteins, osteocalcin, and other components of the bone matrix known as osteoid. The osteoblasts accumulate at the periphery of the ossification center and continue to secrete osteoid at the center of the nodule. As the process continues, the osteoid undergoes mineralization and the entrapped osteoblasts become osteocytes. Within the bony matrix, osteocytes increasingly separate from one another as more matrix is produced, but they remain attached by these cytoplasmic processes. With time, the matrix becomes mineralized and the interconnecting cytoplasmic processes of osteocytes are contained within canaliculi. Now, Initially, newly formed bone matrix appears as small, irregularly shaped spicules and trabeculae. A number of the progenitor cells come into apposition with the initially formed spicules and become osteoblasts and add more matrix. This process is called oppositional growth. By this process, the spicules enlarge and become joined in a trabecular network. Through continued mitotic activity, the osteoprogenitor cells maintain their numbers and thus provide a constant source of osteoblasts for growth of the bone spicules. The new osteoblasts in turn lay down the bone matrix in successive layers and give rise to oven bone. Now, the interconnecting spaces are occupied by connective tissue and blood vessels. Further growth and remodeling results in replacement of oven bone by compact bone in the periphery and spongy bone in the center of the newly formed bone. Spaces between trabeculae become occupied by bone marrow cells that arrive with blood vessels. Bone tissue formed by this process is referred to as membrane bone or intramembranous bone. So this is all about the intramembranous mode of ossification. If you have any question, please comment. If you like the video, then share. Subscribe the channel for more new videos. Stay with us. Keep watching. Thank you.